Picture this. You're standing in front of a 2,000-liter bioreactor worth $500,000. Inside are billions of cells producing a life-saving cancer drug. You reach for the control panel to adjust the oxygen flow rate, and suddenly, the entire batch crashes. $20 million of product just gone. And even more importantly, critical patient drug products that would help treat thousands of patients that will now be harder to come by for patients to get access to over the next year. Your heart sinks, but then you just reset the process because this was all happening using a simulated virtual bioreactor on a computer screen. The cells were simulated, the bioreactor was digital, but the lesson, that was absolutely real. Now, before you think this is just another tech buzzword episode, let me tell you why this matters to you. Whether you're sipping your morning coffee made from precision fermented beans, taking biologic medicines produced in bioreactors, or wearing clothes made from biofabricated materials, bioreactors as the central manufacturing platform in the biotechnology century will touch nearly every aspect of your life. And the people that will operate these biological factories, they will need to be trained on the best tools available. That's where our story begins. Welcome to Apprentice.io's Industry Insights. I'm Frank Maggiore, and today we're diving into the world of virtual bioreactors, the technology that's revolutionizing how we train scientists, optimize processes, and maybe, just maybe, democratizing access to biotechnology education worldwide. This topic is very personal to me because I was in charge of the pioneering Biostat T project when I worked for Satori Steam Biotech about a decade ago. So what is the Biostat T? The T stood for training, and it all started with a simple observation. When we were asking universities to train the next generation of bioprocess engineers on either very expensive new equipment, ancient barely working legacy equipment, and equipment that required extensive manual cleaning of yeast cultures at the end of the process, all of which were either not practical, ruined hands-on experience for students, or made teachers and teaching assistants miserable. Not exactly practical, right? Also, the learnings around fermentation and cell culture processes don't fit neatly into a 1-hour lab session. But here's where it gets interesting. What if we could create a perfect digital replica of those bioreactors? Not just a simple simulation, but a comprehensive virtual environment where students could make mistakes, experiment with parameters, watch their virtual cells grow and die based on real biological principles. What if we can compress months of fermentation into hours of training? What if we could let a student in rural Iowa operate the same virtual bioreactors as students in Mumbai? The idea around this was to transform not just education of how to use bioreactors, but how we develop new drugs, optimize production processes, and even predict when a batch might fail before it actually does. We're talking about a technology that bridges the gap between the theoretical knowledge in textbooks and the practical skills needed in multi-billion-dollar biomanufacturing facilities. Today, we'll explore how virtual bioreactors differ from their physical counterparts, scale from tiny research vessels to massive production tanks, integrate with modern manufacturing execution systems, and might just hold the key to training the millions of bioprocess professionals that are critical to nearly everything we do in the biotechnology century. Let me paint you a picture of traditional bioreactor training, and I promise it's not pretty. A professor at a major university has 120 bioengineering students. Their lab only has 3 ancient bench-scale bioreactors. Each run takes 2 weeks. Do the math, and you see why most students graduate having touched a real bioreactor maybe twice. But it gets worse. Remember that $500,000 commercial-scale bioreactor I mentioned earlier? That's actually on the cheaper side for industrial-scale equipment. Add in the cost of media, we're talking thousands of dollars per run, plus the cells, the utilities, the maintenance, and suddenly you understand why most universities make do with bench-scale systems that bear about as much resemblance to industrial bioreactors as a toy plane does to a Boeing 747. And then there's the safety dance. Ever see what happens when someone accidentally contaminates a bioreactor? It's not just the lost product, it's the cleaning, the investigation, the documentation. In industry, that's a learning experience. In academia, that's a semester-ending disaster. This is where virtual bioreactors flip the script entirely. Imagine giving every student their own bioreactor. No, scratch that. A fleet of their own bioreactors. Want to see what happens when you spike the pH to 10? Go for it. Curious about oxygen starvation? Try it out. Yeah, kill all those cells. You're such a rebel. Breaking the law, breaking the law. Wonder what contamination looks like in real time? Introduce some virtual mold contamination and watch the chaos unfold. The time compression factor alone is revolutionary. A typical CHO cell culture for monoclonal antibody production runs for 14 to 21 days. In the virtual world, you can run that in 30 minutes. More importantly, you can run it 50 times with different parameters, building an intuitive understanding of cause and effect that would take years to develop in the real world. Professor Volker Haas at Furtwangen University and University College London, one of the pioneers in this field and the brains behind the complex software algorithms used for the Biostat-T unit, puts it brilliantly. Students using virtual bioreactors make all their expensive mistakes for free. His Bioprocess trainer system has shown that students who train virtually perform significantly better when they finally work with real equipment. They're not intimidated by the control systems because they've seen them before. They've learned the menu navigation, they understand the lag between making an adjustment and seeing its effect because they've experienced it dozens of times. But here's what really excites me: accessibility. That student in rural Iowa I mentioned, they can now access the same quality of training as someone at MIT. A community college can offer bioprocessing courses without building $1 million facility. Developing countries can leapfrog infrastructure limitations and train their workforce for the new bioeconomy. The virtual approach also enables something impossible in traditional training: failure analysis. When a virtual batch crashes, you can rewind, replay, and understand exactly what went wrong. Try doing that with 2,000 liters of actual cell culture. This forensic capability transforms failures from disasters into powerful learning opportunities. And let's talk about contamination, the boogeyman of bioprocessing. In the real world, contamination means stopping everything, cleaning, investigating, and often never really knowing what went wrong. In the virtual world, you can intentionally contaminate, watch it spread, see how it affects metabolism, and learn to recognize the early warning signs. It's like a flight simulator for bioprocessing. You can practice emergency procedures without experiencing any actual emergencies. Now let's talk about one of the biggest headaches in bioprocessing: scale up. It's the art and science of taking something that works perfectly in a 2-liter flask and making it work in a 2,000-liter production vessel. Sounds simple, it's anything but. Here's the thing about scaling up: the cell strains are identical and the biology might stay the same, but literally everything else changes. The physics, the mixing patterns, the gas transfer rates, the shear forces. It's like trying to recreate your grandmother's cookie recipe, but instead of doubling it, you're making 1,000 times more in a different oven with industrial grade equipment. Virtual bioreactors are revolutionizing how we approach this challenge. They use sophisticated mathematical models. We're talking computational fluid dynamics, metabolic flux analysis, and machine learning algorithms to predict what happens as you scale up. Let me give you a concrete example that'll blow your mind. In a 2-liter bioreactor, your mixing time might be a few minutes. Scale that up to 2,000 liters using traditional rules of thumb, and suddenly your mixing time is close to half an hour. For bacteria, that's an eternity. In that time, cells at the bottom might be oxygen starved, while cells at the top are practically hyperventilating. Virtual bioreactors can model these gradients, showing you exactly where the dead zones will be and how to modify your impeller configuration to fix them. The oxygen volumetric mass transfer coefficient, KLA for the nerds in the audience, is particularly tricky. In small scale, you might achieve a KLA of 200 per hour easily. At production scale, good luck hitting 50 without turning your cells into smoothies from the shear forces. Virtual systems let you experiment with different sparger designs, bubble sizes, and agitation rates without wasting a single cell. Professor Haas's team has created virtual models spanning from 60 milliliter microbioreactors all the way up to 20,000 liter production vessels. The magic is in the middleware, the mathematical translations that account for changes in surface area to volume ratios, hydrostatic pressure, and even the way that bubbles coalesce differently at different scales. Here's where it gets really cool: parameter scaling. Let's say you need to maintain the same power input per unit volume across scales, a common scale-up strategy. In the virtual world, you can instantly calculate that your 100 RPM in a 2-liter vessel translates to 23 RPM in a 2,000-liter vessel. But wait, that slow stirring speed won't suspend your cells properly, so you try scaling by impeller tip speed instead, which gives you 45 RPM. Better, but now your power consumption is through the roof. This is the kind of optimization that traditionally takes months of pilot scale runs, each costing hundreds to thousands of dollars. In the virtual world, it's an afternoon's work. And let's not forget about the equipment differences. Your lab-scale bioreactor might use a simple Rushton turbine impeller. Production scale, you're looking at multiple impeller types, maybe a combination of radial and axial flow patterns. The virtual environment lets you test combinations that would be prohibitively expensive to prototype physically. The real game changer is being able to simulate equipment you don't even own yet. Considering that new 4,000-liter single-use bioreactor, run your process virtually first. See how your cells will behave, identify potential problems, optimize your control strategies, all before spending a dime on actual equipment. When Sartorius launched the Biostat-T virtual bioreactor training tool, it was like introducing the iPhone in a world of flip phones. And there's a running story in my life for being too far ahead in technologies before the market is ready to accept them. The Biostat-T was revolutionary, way ahead of its time, and ultimately discontinued. But like all good failures, it taught us invaluable lessons. The Biostat-T Cho was brilliant in its simplicity. It mimicked the exact interface of the Biostat STR 200-liter bioreactor, complete with real cultivation data from actual Cho cell runs. Students could practice everything from basic menu navigation to complex fed batch strategies. It was free. It only required a basic computer, and it could prevent the kind of mistakes that cost companies millions of dollars. So why did it fail? Well, fail might be too strong of a word. Let's say it was a solution waiting for the market to catch up to it. First, timing. When the Biostat-T launched, virtual bioreactor training was still science fiction for most people. Cloud computing was just in its infancy, and the idea of digital twins was confined to aerospace engineering. The bioprocessing industry, traditionally conservative, wasn't ready to trust virtual training for real-world applications. Second, the ecosystem challenge. A virtual bioreactor in isolation is like a smartphone without apps. Useful, but not transformative. The Biostat-T needed integration with learning management systems, competency assessments, and industry certification programs. It needed a community of users sharing scenarios and best practices. None of that infrastructure existed yet. But here's what the Biostat-T got absolutely right, and why its DNA lives in today's solutions. It proved that students trained virtually could outperform those trained traditionally. It demonstrated that complex biological behaviors could be accurately simulated. Most importantly, it showed that the industry was hungry for better training solutions. They just needed more than a standalone tool. The discontinuation of the Biostat-T wasn't a failure. The rest of the industry just needed to catch up. Today's solutions, like digital twin platforms from companies like Ansys, build on the foundations that the Biostat-T project laid. So what's going on with the virtual bioreactor landscape? The vision of the Biostat-T, democratizing bioprocess training, making expensive mistakes cheaply, compressing time. It's all happening now, just in ways more amazing than anyone could have imagined. Imagine if your virtual bioreactor wasn't just a training tool, but a crystal ball that could predict the future of your actual production batches. Welcome to the world where digital twins meet manufacturing execution systems. It's like giving your bioreactor a highly intelligent twin sibling who can see into the future. Let me break this down with a real scenario. You're running a $20 million batch of monoclonal antibodies in a 2,000-liter bioreactor. Your Apprentice.io tempo manufacturing cloud is collecting data every second. pH, dissolved oxygen, temperature, metabolite levels, cell density. Now here's where the magic happens. That data can be fed into a digital twin running in parallel, simulating your exact process in real time. 3 days into your 14-day batch run, the digital twin notices something. The lactate production is 5% higher than normal. Not alarming yet, but the digital twin runs 1,000 scenarios in the cloud. In 73% of them, this leads to an early cell death by day 10. The MES alerts you, potential batch failure predicted. Recommends action, reduce glucose feed rate by 15% for the next 24 hours. You make the adjustment, the batch is saved. $20 million preserved by a combination of real-time data and predictive modeling. This isn't science fiction, it's happening in biomanufacturing facilities right now. But it goes deeper. The MES isn't just collecting data, it's learning. The system now knows that in your specific bioreactor with your specific cell line, it's an early warning sign. The digital twin becomes smarter, more predictive, and more valuable with every run. Here's where it gets absolutely wild. Scenario testing. Let's say you're considering switching from batch to perfusion mode for continuous manufacturing. Traditionally, that's months of pilot studies and millions of dollars in investment. With an MES integrated digital twin, you can run the entire transition virtually first. Test different perfusion rates, cell retention devices, media formulations, all without touching your actual production line. AI integration is the secret sauce. The system doesn't just follow pre-programmed rules, it learns patterns humans might miss. Maybe it notices that batches started on Tuesdays have 3% higher yields. Turns out, that's when fresh media is cleared by quality control microbiology. Or it identifies that a specific operator's batches consistently perform better. They have a gentler touch when adjusting the parameters. These insights, impossible to catch manually, become obvious when AI analyzes thousands of data points across hundreds of different batches. And then there's the step-by-step guidance revolution. New operator, the MES-connected digital twin becomes their personal coach. Based on current conditions, navigate to menu, process control, agitation, increase stirrer speed to 125 RPM for 10 minutes to improve oxygen transfer. It's like having an expert looking over your shoulder, except this expert has analyzed every batch ever run in your facility. For process development, this integration is transformative. You can use historical data from your MES to train digital twins for new products. Starting development of a new biosimilar, your digital twin already knows your equipment's quirks, your operator's patterns, your facility's capabilities. It's like starting with a decade of experience built in. The continuous improvement loop this creates is beautiful. Real batches generate data. Data trains digital twins. Digital twins predict optimizations. Optimizations improve real batches. It's a virtuous cycle that gets more powerful with every iteration. Let's zoom out and look at the big picture, how virtual bioreactors are transforming every stage of the biomanufacturing life cycle. From the moment a scientist dreams up a new biological product to the day it rolls off the production line, virtual bioreactors are accelerating timelines and reducing risks in ways that seemed impossible just a few years ago. Let's start with process development. Traditionally, developing a bioprocess is like solving a Rubik's Cube blindfolded. You have hundreds of variables, media components, feeding strategies, temperature profiles, pH set points, and you need to find the combination that maximizes yield while maintaining quality. Each experiment takes weeks and costs thousands of dollars. Enter virtual bioreactors with design of experiments on steroids. Instead of testing 10 conditions physically, you can test thousands of them virtually. Machine learning algorithms identify patterns and optimal ranges. Then you validate only the most promising conditions in real bioreactors. What used to take years now can take a few months. Tech transfer, moving a process from development to manufacturing, is where virtual bioreactors really shine. It's bioprocessing's version of the works on my machine problem. Your process runs perfectly in the development lab's 50-liter bioreactor. Will it work in manufacturing's 2,000-liter vessel? What if you decide to get a 4,000-liter vessel? Virtual commissioning gives you the answer before you transfer a single cell. Continuous manufacturing, especially perfusion processes, is where virtual bioreactors become absolutely essential. Perfusion is like keeping a plane in the air for months. Everything needs to work perfectly all the time. Virtual bioreactors let you test failure scenarios safely. What happens if a pump fails? If the cell retention device clogs? If the perfusion rate drifts? You can practice recovery procedures, optimize alarm settings, and train operators without risking a real batch. The golden batch concept, that mythical perfect run everyone tries to replicate, becomes achievable with virtual bioreactors. By analyzing all of your historical data, identifying what made your best batches special, and then using virtual runs to test hypotheses, you can reverse engineer success. You may discover that golden batches all have a specific dissolved oxygen profile during the growth phase, something that you would never notice in years of production with paper batch records. Virtual testing confirmed it, and now every batch can be a golden batch. Scaled down models, the unsung heroes of bioprocessing, get supercharged by virtual technology. Need to investigate a deviation that happened at the 2,000-liter scale? Create a virtual scaled down model that accurately represents the large-scale conditions in a 2-liter format. Test fixes virtually, validate the best ones physically, implement at scale. It's troubleshooting at the speed of thought. Process optimization never stops, and virtual bioreactors make it continuous. Every real batch generates data that refines the virtual model. The virtual model suggests improvements. It's like having a process development team working 24/7, constantly finding ways to squeeze out another percent of yield or reduce costs by another dollar per gram. The education revolution happening with virtual bioreactors is nothing short of extraordinary. Universities worldwide are transforming from equipment-limited institutions to innovation powerhouses, and the industry is taking notice. At University College London, Professor Frank Begans has integrated virtual bioreactors into the core curriculum. Students start with virtual runs in year 2, learning the basics without the pressure of potentially breaking expensive equipment. By the time they touch a real bioreactor in year 3, they're confident and competent. But it's not just about avoiding mistakes. Virtual bioreactors enable pedagogical approaches, impossible with physical equipment. Want to teach the importance of dissolved oxygen control? Create a scenario where the dissolved oxygen probe fails mid-run. Students must diagnose the problem from secondary indicators and implement workarounds. It's problem-based learning on steroids. Additional multiplayer capabilities are game-changing. At Linköping University in Sweden, students operate a virtual bioethanol plant together. One team manages fermentation, another handles separation, a third optimizes distillation. They must coordinate, communicate, and optimize the entire process as a system. It's like a Bioprocess MBA compressed into a semester. Industry adoption is accelerating for one simple reason: ROI. When a new $10 million bioreactor arrives at a facility, every day of commissioning costs money. Operators practice startup procedures, learn the control systems, and troubleshoot common issues before the equipment even arrives. The competency assessment capabilities are transforming how we certify bioprocess professionals. Instead of paper exams, operators demonstrate skills in virtual scenarios. Can you recover from a failed pump event? How quickly can you optimize a fed batch strategy? Do you follow proper sparger parameters, even when rushed? Virtual assessments are objective, comprehensive, and directly relevant to job performance. Remote training has become essential, and virtual bioreactors deliver. During the pandemic, bioprocessing education could have ground to a halt. Instead, it accelerated. Students accessed virtual bioreactors from home, ran experiments at midnight if they wanted, and collaborated with peers globally. The University of Queensland partnered with institutions in India and Brazil, giving students international collaboration experience without anyone boarding a plane. Industry is also using virtual bioreactors for cross-training. An antibody manufacturer can train their operators on microbial fermentation processes. A food company entering cellular agriculture can prepare their workforce without building pilot facilities. It's breaking down the silos that traditionally separated different bioprocessing sectors. The continuous education aspect is perhaps most exciting. Bioprocessing evolves rapidly. New cell lines, new modalities, new regulations. Virtual bioreactors enable just-in-time training. New gene therapy process, deploy a virtual version for training before the first batch. Regulatory change requiring new documentation, practiced in the virtual world where mistakes don't trigger FDA letters. The data from these educational applications is staggering. Students trained with virtual bioreactors show better retention of complex procedures. They make fewer errors in their first real bioprocess runs. Most remarkably, they show increased creativity in problem solving, likely because they've been able to experiment freely in virtual environments. As we stand at the threshold of what many are calling the biotechnology century, virtual bioreactors represent more than just a training tool or optimization platform. They're a democratizing force that could reshape the global bioeconomy. Imagine a future where every high school student has access to a virtual bioreactor on their laptop, where a teenager in Bangladesh can develop the same intuition for bioprocessing as one in Boston, where brilliant minds aren't held back by lack of access to expensive equipment. This isn't just about education, it's about unlocking human potential on a global scale. The convergence we're witnessing, AI, digital twins, cloud computing, and biotechnology, is creating possibilities we're only beginning to grasp. Virtual bioreactors that can design their own experiments, optimize processes autonomously, and even suggest entirely new approaches to production. We're moving from simulation to innovation, from digital twins to digital pioneers. The economic impact is staggering. Every failed batch prevented saves millions. Every day shaved off process development accelerates life-saving treatments to patients. Every operator trained virtually rather than physically reduced costs that could be redirected to research. We're not just making bioprocessing more efficient, we're making it more accessible, more innovative, and more impactful. But here's what gives me the most hope, the sustainability angle. Virtual bioreactors let us optimize processes for minimal resource use, testing strategies for reducing water consumption, energy use, and waste generation, all without consuming actual resources. As we face climate challenges and resource constraints, this capability becomes not just valuable, but essential. The next generation of bioprocess engineers won't see virtual and physical bioreactors as separate entities. They'll see them as complementary tools in an integrated ecosystem. Physical bioreactors generate data, virtual bioreactors explore possibilities, and the cycle continues, each iteration bringing us closer to perfect processes. For those of you listening who work in bioprocessing, the message is clear. Embrace these tools. They're not replacing your expertise, they're amplifying it. Your knowledge of biology, your intuition for process behavior, your creative problem solving. Virtual bioreactors make all of these more powerful. For educators, the opportunity is transformative. You can now offer world-class bioprocessing education regardless of your budget or location. Partner with others, share scenarios, and build a global community of practice. The barriers that once limited bioprocess education are crumbling. For students and aspiring bioprocess engineers, you're entering the field at one of the most exciting times in its history. The tools available to you would have seemed like magic to previous generations. Use them. Experiment fearlessly in virtual worlds. Build intuition through thousands of virtual runs. When you enter the workforce, you'll be prepared for challenges we can't even imagine yet. And for humanity, virtual bioreactors are helping us to build the infrastructure for the bio-based economy. From cell-cultured meat to sustainable materials, from personalized medicines to carbon-negative chemicals, all of these require sophisticated bioprocessing. Virtual bioreactors make this future achievable by making bioprocess expertise scalable. As we close, I want to leave you with this thought. We're not just virtualizing bioreactors, we're democratizing the ability to harness biology for human benefit. Every virtual batch run, every student trained, every process optimized brings us closer to a world where biological manufacturing is as accessible and impactful as information technology became in the last century. The biotechnology century isn't coming, it's already here. Virtual bioreactors aren't just tools for this new era, they're the training grounds where we'll prepare the millions of bioprocess professionals needed to build a sustainable, healthy, and prosperous future for us all. So whether you're a seasoned professional, a curious student, or someone who simply cares about the future, remember this. In the virtual world of bioprocessing, every mistake is a lesson, every experiment is possible, and every person has the potential to contribute to the biological revolution that will define our century. In an industry where precision and compliance are paramount, Apprentice's Tempo platform offers a cutting-edge manufacturing execution system solution. It's cloud-based, mobile-first, operator-centric, with the most intuitive UI in the industry, making it perfect for today's dynamic pharmaceutical manufacturing environments and for the production of the world's most complex drug products. Whether you're working on groundbreaking cell and gene therapies, biologics, or small molecules, Tempo provides a fully validated MES system that's both powerful and easy to use. It streamlines your processes, ensures compliance, and adapts to your specific manufacturing needs. So get off of paper batch records and discover how Apprentice's Tempo Manufacturing Cloud can transform your operations by visiting us at apprentice.io. Attention industry professionals, revolutionize your manufacturing process with the Apprentice Tempo Manufacturing Cloud, a manufacturing execution system built to streamline your operations, boost efficiency, and ensure compliance all in one powerful platform. Experience the future of manufacturing with our free 30-day trial. Scan the QR code on your screen now to unlock a world of innovation. Don't just keep pace, set the tempo as an industry leader with Apprentice. That's all for Apprentice.io's Industry Insights. I'm Frank Maggiore. Thanks for listening. Be sure to like and subscribe. Hit the bell icon for notifications when new episodes drop. Please comment on if you have any additional topics you would like for me to cover in this space, and we'll see you next week for more Industry Insights.